to another lecture series on Enterobacteriaceae. Today we are looking at two interesting closely related members of Enterobacteriaceae by name Salmonella and Shigella. These two bacteria are closely related in terms of their sources, in terms of disease they cause, and also in isolation. First of all, we look at Salmonella. Genus Salmonella. The genus Salmonella are typically non-spore forming bacteria. They are predominantly motile with peritrichous flagella. They are chemo-organotrophs and facultative anaerobes. And we have three notable species of Salmonella which are widely recognized, although two are the most medically relevant species. The first species is Salmonella enterica. Salmonella enterica is the most diverse and clinically relevant species. It has about 2,500 serotypes, which can also called serovars, which are linked to several illnesses. Example includes Salmonella typhi, which is responsible for typhoid fever. We also have Salmonella typhimurium, which is responsible for severe gastroenteritis. So the second species is Salmonella bongori, though Salmonella bongori is less common than Salmonella enterica, though in some certain environments it can be found in cold-blooded animals. Salmonella bongori is not typically associated with human disease like that. Then the third one is Salmonella subterranea. Now, Salmonella subterranea is one of the rare species which does not have much information on it with regards to distribution, with regards to its potential for causing disease. However, among these three, Salmonella enterica constitutes major pathogenic species and are widely known all over the world. Interestingly, the two main species, enterica and bongori, were previously grouped together under a single species called Salmonella enterica. However, due to genetic and phenotypic differences, they were classified as separate species in 2005. It is also important to note that even with the three species, a number of serotypes within enterica, Salmonella enterica makes Salmonella a highly diverse and complex genus. So each serotype has unique characteristics and different potentials to cause infection in humans and animals. So the genus, like I said, has about 2,600 serotypes, each with unique antigenic properties. Some of the common and clinically significant ones have been explained previously, and Salmonella enterica subspecies responsible for human salmonellosis cases worldwide, while typhi causes typhoid fever, and paratyphi A and B they also cause similar typhoid fever, and Salmonella typhimurium cause foodborne illness. So these species and subspecies of Salmonella are those that are medically important under the genus Salmonella. They are commonly found in the habitat. They are commonly found in animals, poultry, cattle, pig, and other warm-blooded animals that harbor them without showing any symptoms. When they get into human gut through contaminated food or water, or through the consumption of this animal, they can cause disease infection. Then in the environmental reservoir, we find them abundant in the soil and also in the water, and this is usually due to the fact that some certain environments have disposed fecal contaminated sewages that run unfiltered to the water system, making salmonella abundant in the water system. Then in food sources, they are abundantly present in undercooked or raw meat, poultry, eggs, dairy products, and so on. So these are sources of salmonella infection, and also area where salmonella can be isolated for investigation purpose. In terms of biochemical characteristics, salmonella are motile, they are flagellated, they are non-lactose fermented, and they produce hydrogen sulfide which appear as a black colony on media, especially on indicator media that differentiate them. Example of such indicator media that differentiate them is Salmonella Shigella area that allow them to grow with a color change of black colony. They also produce urease, and Salmonella can be grown on selective media that suppress other organisms. Example is Maconche, Salmonella Shigella agar, Ectoin, enteric agar, which are selective for Salmonella and suppress the growth of other similar organisms in the environment. Now, Salmonella enterica is classified into 6 subspecies. It will be prudent to explain what subspecies mean in bacteria. Subspecies is a taxonomic rank below a species. It is used to classify populations of bacteria within a species that has some distinct characteristics that is different from the main species, but are still similar enough to interpret and be considered part of the same species. So because the species is actually the basic unit of every biological classification in bacteria. When a group of bacteria share similar characteristics and can interbreed to produce a fertile offspring, while a subspecies is a subdivision of a species that exhibits some differences from the main group in terms of morphology, sizes, arrangement of cells, physiology, metabolic activity, production of toxins, and tolerance in the environment. So, subspecies of Salmonella enterica are Salmonella enterica, Salame, Arizona, Garizone, Uteni, and Indica. And a typical subspecies of Salmonella could be written as Salmonella enterica subspecies Arizona or Salmonella enterica subspecies Indica. So, the subspecies of Salmonella are into 6 divisions. Salmonella enterica also has more than 2,500 serovars. A serovar, also known as a serotype, is a type of variation within a bacterial species that differ from other variations based on surface antigens. So these antigens are molecules on the bacterium outer surface that trigger an immune response in the host by identifying the specific serovar of bacterium. So scientists can actually gain valuable information about its pathogenicity, transmission, and antibiotic resistance based on different serovar. So Salmonella enterica serovar are defined based on their somatic O liposaccharide and flagella H antigen. So the full name of the serovar is given, for example, Salmonella enterica subspecies enterica serovar typhimero, and can be abbreviated as Salmonella typhimero. Identification and isolation of Salmonella can be obtained First of all, it can be isolated from food, it can be isolated from animal sources, it can be isolated from egg, dairy products, vegetables, herbs, spices, and other contaminated areas of the bathroom and toilet. These are very sources of isolation of Salmonella for identification in the laboratory. So on cultivation of Salmonella, the clinical samples can be cultured directly on selective agar such as XLD, xylose lysine deoxycholic agar. However, if a stool sample is the source of isolation, then the Salmonella culture have to first of all be enriched to suppress the growth of other organisms. An example of an enrichment broth for Salmonella is selenite cysteine broth, which suppresses the growth of other organisms present in the stool and can support the growth of Salmonella. Then it can be subcultured in the selective agar such as Salmonella Shigella agar. Salmonella will produce a black colony as seen on this picture because it usually interacts with hydrogen sulfide to form a black colony. This is a differentiating colony for Salmonella and Shigella because both of them can be isolated with this same SS agar. However, their colony are differentiated by the production of black colony as a result of hydrogen sulfide hydrolysis. Salmonella Shigella, Salmonella economic importance is notably observed in terms of outbreak of foodborne disease, which can be known as salmonellosis, and these are associated with dairy products, food contaminated, um, confectionaries, cooked and undercooked meats, ice cream, and so on. And the symptoms of this salmonellosis include gastroenteritis, vomiting, nausea, abdominal cramps. It can also produce enteric fever and sepsis, which can be life-threatening in vulnerable individuals. It can also cause typhoid fever and also cause some other similar infection. So, Salmonella is one of the notable members of Enterobacteriaceae that is associated with infection, commonly foodborne infection. So now we'll look at the next close related species, genus, which is Shigella. The genus Shigella is also an interesting member of Enterobacteriaceae. The genus consists of notable members which are related to Salmonella. Both of them are closely related, but there are differences in terms of the fact that Salmonella are primarily transmitted through contaminated food, such as poultry, eggs, unpasteurized milk and water, and they can also spread through contact with animals or the environment, while Shigella are mainly transmitted through person-to-person contact, especially in places with poor hygiene. Housefly can also play a role in transmission of Shigella. In terms of motility, both of them are differentiated because Salmonella is actually motile and it has a tiny flagella, while Shigella is not motile. However, both of them are similar in terms of fermentation. Both are non-lactose fermenter, but Salmonella produces hydrogen sulfide on some culture media, which makes this colony to be black, whereas Shigella does not produce such colony. Salmonella usually causes diarrhea, vomiting, abdominal cramp. When I say diarrhea, watery diarrhea, abdominal cramp, and this can be self-limiting in many cases, but may progress to more serious infection if care is not taken. Whereas Shigella causes bloody diarrhea, amphibia, and severe abdominal cramp, and can lead to complications such as dehydration and malnutrition in terms of symptoms. So these are uh, differences between the two genus, even though they are closely related. Shigella is classified into different serogroups. Serogroup A, which consists of Shigella dysentery, it has about 15 serotypes. This group includes the most virulent Shigella species, such as Shigella dysentery type 1, and it produces a toxin called Shiga toxin and can cause severe dysentery and death. As a matter of fact, Shigella dysentery is closely related to Shiga toxin E. coli, also known as E. coli 0157, because of the fact that they both produce this Shiga toxin. And also, we also have serogroup B, which is Flexneri, that has about now 9 serotypes. They often cause watery or bloody diarrhea, and this is very common in developing countries. The serogroup C, which is known as Shigella boydii, has about 19 serotypes, though this is less common than Flexneri, but also still cause significant illness in some regions. The last one, which is Shigella sonnei, consists of one serotype and is the most common Shigella species worldwide, especially in developed countries, usually cause milder illness than other serogroups. Yeah. Shigella can be found in habitat primarily in human intestinal tract, specifically the colon. They are not free-living bacteria and cannot survive outside the host for extended period of time. That is why they are not very common in the environment like Salmonella. Their transmission is through fecal-oral route, either via direct contact with an infected person or via contaminated food and water. So the habitat of Salmonella is not as widely distributed like the habitat of Shigella is not as widely distributed as the habitat of Salmonella, because Shigella is a bit restricted while Salmonella can be widespread. The biochemical characteristics, some of which I've said before, but they are non-motile, they are facultative anaerobic, they are non-lactose fermental, urease positive, and lysine decarboxylase positive, which differentiate them from Salmonella on certain media. Shigella can be isolated using the selective agar known as Salmonella Shigella agar, also used for isolation of Salmonella. Now, the difference is that Shigella produces a transparent or translucent colony, which means the colony can blend with the media, with the agar, whereas Salmonella will produce a black colony. Also on McConkey agar, it produces a pink translucent colony, while in McConkey lactose agar, it produces a convex colony, and also on XLD, it produces a transparent or red colored agar, colony on the agar, that is for Shigella. In terms of economic importance, Shigella is known to cause acute severe diarrhea, and it normally produces watery, often contaminated with with blood and mucus. Those are the symptoms, and before the individual goes to stooling, it manifests in terms of abdominal cramps and pain, and also can provide elevated fever, nausea, and vomiting. And other complications can arise whereby the patient can lose water if restoration of electrolyte is not administered on time, may lead to more complication. It's a public health body in terms of cost of treatment and hospitalization. Shigella causes a lot of illness among caregivers and decreases tourism and food export in affected region because of its severe health burden. And in terms of global impact, shigellosis is a major public health concern, particularly in developing countries with poor sanitation and hygiene practices because of person-to-person transmission of Shigella. So areas where there is poor hygiene, people can easily contaminate one another, especially when they are harboring this pathogen in their hands and so on. So Shigella also presents itself as emerging drug-resistant bacteria, which pose serious challenge to treatment and control. And this is a global challenge and global problem. But this can be mitigated by the use of appropriate antibiotics that can inhibit the growth of Shigella. So there are practice questions which I would like you to uh, take a look at and can guide you into further reading. Mind you, some of the questions in this practice question may not be totally reflected in this lecture. However, it may require you to do some additional reading. That is the purpose of this practice question. So enjoy.